Good morning, students. I, myself, Dr. Vasantrao B. Kadam, Associate Professor, Department of Botany, KTHM College, Nashik. Students, I have completed my M.Sc and PhD in Botany, specialty in plant physiology. I am a research guide for M.Phil and PhD in the subject of botany and environmental science of Dr. Babasaheb Ambedkar Marathwada University, Aurangabad and Savitribai Phule Pune University, Pune. 4 students are completed PhD and 4 students are working for PhD under my guidance. I have published 145 research papers in various national and international journals having good impact factors. I also published 10 books for UG, PG and research students. I am a member of editorial board of 20 national and international journals. Students, I teach you the paper number 3. Title of this paper is Plant Physiology. Now today we are going to learn the structure of chloroplast and various functions of chloroplast related to photosynthesis. Students, you know the basic definition of photosynthesis. Now, photosynthesis means synthesis of food material by the green cells of plants, that means chlorophyll pigments, in presence of sunlight with the help of carbon dioxide and water. That means 4 things are required for the synthesis of food: chlorophyll pigments, sunlight, water and carbon dioxide. Now, photosynthesis occurs on thylakoid membrane. Chloroplasts are bounded by 2 membranes, outer membrane and inner membrane, which do not contain chlorophyll and do not participate directly in photosynthesis. Of these 2 membranes, the outer one, like the outer mitochondrial membrane, is permeable to metabolites of small molecular weight. It contains proteins that form very large aqueous channels. The inner membrane, conversely, is the permeability barrier of the chloroplast. It contains transporters that regulate the movement of metabolites into and out of the organelle. Unlike mitochondria, chloroplasts contain a third membrane, the thylakoid membrane, that is the actual site of photosynthesis. In each chloroplast, the thylakoid membrane is believed to constitute a single interconnected sheet that forms numerous small flattened vesicles, the thylakoids, which commonly are arranged in the stalks termed grana. The spaces within all the thylakoid constitute a single continuous compartment, the thylakoid lumen. The thylakoid membrane contains a number of integral membrane proteins to which are bound several important prosthetic groups and light absorbing pigments, most notably chlorophyll. Carbohydrate synthesis occurs in the stroma. The soluble phase between the thylakoid membrane and the inner membrane. In photosynthetic bacteria, extensive invaginations of the plasma membrane form a set of internal membranes, also termed thylakoid membranes or simply thylakoids, where photosynthesis occurs. Students, now look at the pictures. Now the first picture is the structure of internal structure of leaf. Now leaf having cuticle, upper epidermis, mesophyll cells, lower epidermis and cuticles. A number of chloroplasts are present in the mesophyll cell. Now this is the structure of chloroplast where actual photosynthesis occurs. Now the complete process of photosynthesis is divided into two parts. One is known as light dependent phase and second one is known as light dependent phase. Now this is the portion of grana. Now in grana portion the light dependent phase of photosynthesis occurs. That means all the stages of light dependent phase occurs in grana portion of chloroplast. And now this is the stroma portion of chloroplast where the dark reaction, that means light independent phase of photosynthesis occurs in the stroma portion. Now in the stroma portion, enzymes that catalyze carbon dioxide fixation and start synthesis, actually glucose synthesized in the dark portion of chloroplast is known as stroma. Now this is the structure of granum. The thylakoid lumen is present in each grana. Now this is useful for photophosphorylation and also the pumping of protons towards the membrane of thylakoid from lumen. Three of the four stages in photosynthesis occurs only during illumination. That means out of four, three processes are during illumination. It is convenient to divide the photosynthetic process in plants into four stages, each occurring in a defined area of the chloroplast. Number one, absorption of light. Number two, electron transport leading to the reduction of NADP to NADPH. Number 3, generation of ATP by cyclic and non-cyclic photophosphorylation, and conversion of CO2 into carbohydrates, that means carbon fixation. All four stages of photosynthesis are tightly coupled and controlled so as to produce the amount of carbohydrates required by the plants. Now, stage first, second, and third required light and chlorophyll pigments. And now stage fourth, conversion of CO2 into carbohydrates. It requires carbon dioxide. All the reactions in stage first and third are catalyzed by proteins in the thylakoid membrane. The enzymes that incorporate carbon dioxide into chemical intermediates and then convert it to starch are soluble constituents of the chloroplast stroma. The enzymes that form sucrose from 3 carbon intermediates are in the cytosol. Now first stage is the absorption of light. During daytime, light is absorbed by the plants. In grain cells of plants, number of light harvesting complexes or antenna complexes are present, and now the antenna complex are absorb the specific wavelength of light. Now the wavelength of light is in between 380 to 780 nanometer of light, and 380 nanometer to 780 nanometer of light is known as visible light. That means the light is useful for the process of photosynthesis, that means the synthesis of glucose. The initial step in photosynthesis is the absorption of light by chlorophyll attached to proteins in the thylakoid membrane. Now in thylakoid membrane, number of chlorophylls are there, and chlorophylls having the antenna complex. And now this antenna complex absorb the specific wavelength of light. Like cytochromes, chlorophylls consist of porphyrin rings attached to a long hydrocarbon side chain. This is the long hydrocarbon chain. They differ from cytochromes in containing a central magnesium. This is the magnesium ion rather than Fe atoms, and having an additional 5-membered ring. The energy of the absorbed light is used to remove electrons from an unwilling donor, that means water, in green plants, forming oxygen, and then to transfer the electrons to a primary electron acceptor, a quinone designated Q, which is similar to coenzyme Q. Now students, after absorption of specific wavelength of light, and now the absorbed light transferred by the antenna complex towards the core complex. Core complex is present in chlorophyll, and now this core complex controlling the complete 40 to 60 number of chlorophyll molecules. Now the absorbed solar energy, sunlight, by the antenna complex, transferred towards the core complex, and core complex decide one chlorophyll molecules out of 40 to 60 chlorophyll molecules, and these absorbed light energy core complex transfers towards the chlorophyll molecules. Each and every chlorophyll molecules outer orbit having two electrons. Both electrons are moving opposite directions, and now these absorbed light taken by one electrons, and now these electrons moves from ground level state to the excitation state. Now the electrons taking the solar energy, and we are called the excitation state of the electrons. And now these electrons moves towards the first primary electron acceptor, is known as coenzyme Q or phagocytic. Now chlorophyll A, chlorophyll B and carotenoids are the pigments, photosynthetic pigments present in leaf. They absorb the various wavelengths of light. Now you look at this picture and you observe that the chlorophyll A absorb the light in between 400 to 500 nanometer of light. That means they absorb the blue light. Again, chlorophyll A absorb the light in between 650 to 700 nanometer of light. That means the red portion of the light. Now invisible light, that means 400 to 700 nanometer of light having the different 7 colors, and now this blue color absorb the light by chlorophyll pigments A, and again 650 to 700 nanometer of light during the red portion of light. Likewise, the chlorophyll B also absorb the light in between 400 to 500 nanometer of light in blue color, and again in 600 to 650 nanometer of light in red color. Same, carotenoids also absorb the light between 450 to 500, and now this absorbed light transferred towards the chlorophyll A by the carotenoids. Number 2 is electron transport. Now this electron move from the quinone primary electron acceptor through a chain of electron transport molecules in the thylakoid membrane until they reach the ultimate electron acceptor, usually NADP. It is reducing it to NADPH. The transport of electron is coupled to the movement of protons from the stroma to the thylakoid lumen, forming a pH gradient across the thylakoid membrane. In much the same way that a proton motive force is established across the mitochondrial inner membrane during electron transport. Thus, the overall reaction of stages first and second can be summarized as 2 water molecules plus 2 NADP, some amount of light, it gives 2 protons and reduction of 2 NADPH2 and releases of or evolve of some amount of oxygen. Now during electron transport, what happens in non-cyclic photophosphorylation? The absorption of light by antenna complex, then absorbed light transferred towards the core complex, and then core complex decide the electrons to excite. That means taking this solar energy to move from different cytochrome system and prepare the number of ATPs during the journey of this electron transport. Now side by side during non-cyclic photophosphorylation in pigment system second its reaction center is 680 nanometer. The water molecule is oxidized or break down into protons and electrons, and now these electrons are entered into the electron transport system chain. This is the continuous process, and now the breakdown of water molecules or oxidation of water molecules find out by Van Niels in 1941. Many photosynthetic bacteria do not use water as the donor of electron. Rather, they use molecules such as hydrogen gas or hydrogen sulfide as the ultimate source of electrons to reduce the ultimate electron acceptance, that means NAD rather than NADP. Now the third part is generation of ATP, that means synthesis of ATP during the movement of electrons. Protons move down their concentration gradient from the thylakoid lumen to the stroma. Now see, this is the protons from thylakoid lumen to the stroma portion through the F1 and F0 complex. Now this is F0 complex, this is F1 complex, which couples protons movement to the synthesis of ATP from ADP and inorganic phosphate. Now students, what happens during pigment system cycle? Its reaction center is 680 nanometer of light. After absorption of light by the antenna complex, now this absorbed light transferred towards the core complex. Core complex is present in the leaf, in the chloroplast, and now this core complex decide to hand over this absorbed energy to any one chlorophyll molecules, which is controlled by 40 to 60 number of chlorophyll molecules. Now after deciding the decided chlorophyll molecules taking this radiant energy, and each and every chlorophyll molecules outer orbit having two electrons, one electrons taking this solar energy and excited to move through these different cytochrome systems. Now what happens? These excited electrons moves towards the plastoquinone, then moves towards the cytochrome B6. In between, few amount of energy is released by these excited electrons, and now this released energy is captured by, combined with these protons, it is captured by adenosine diphosphate plus inorganic phosphate. It is already present in leaf and it is converted into ATP, adenosine triphosphate. This is energy rich molecules. And now these energyless electrons moves towards plastocyanine, then pigment system first. Again students here, pigment system second. First, its reaction center is 3700. Same procedure occurs. Absorption of light by the antenna complex, transfer the light towards the core complex, then core complex decides one chlorophyll molecules to excite, and then the electron molecules moves towards the ferredoxine, then ferredoxine reducing substances, and now this NADP combines with one protons to form NADPH2. It is also the energy molecules like ATP. Now students, side by side here oxidation of water molecules occurs. That means breakdown of water molecules into protons and electrons and releases of some amount of oxygen. Now during the photosynthesis, oxygen released, and now this released oxygen comes from water molecules, not from carbon dioxide. And now these synthesized oxygen molecules released towards the atmosphere, and now these electrons coming from the breakdown of water molecules enter into this cytochrome system for preparation of various ATP molecules. Students, see, this is the structure of CF1 particles and CF0 particles present in the chloroplast. Now, last part of this presentation is carbon fixation. Friends, we learn the synthesis of ATPs, and now we are going to learn the synthesized ATPs are utilized for the conversion of atmospheric carbon dioxide to carbohydrates. Now what happens? This procedure occurs in dark phase of photosynthesis. Why it is called dark phase of photosynthesis or light independent phase? Because there is no need of light for the conversion of carbon dioxide to carbohydrates. Now, during stomata open, the atmospheric carbon dioxide enter into the leaf, and this carbon dioxide is converted into carbohydrates. During the second phase of photosynthesis is known as dark phase of photosynthesis. The ATP and NADPH generated by the second and third stage of photosynthesis, that means during the light of phase of photosynthesis. Now students, the complete mechanism of photosynthesis is divided into two parts. The one part, initial part, is known as light dependent phase, and next part is known as first part is known as light dependent phase, and second part is known as light independent phase. Now what happens in light dependent phase? In a single sentence, synthesis of ATPs. And now what happens in the second phase is known as light independent phase. In a single sentence, that means atmospheric carbon dioxide is converted into carbohydrates. And now we are going to study the conversion of CO2 to carbohydrates. The ATP and NADPH generated by the second and third stage of photosynthesis provides the energy and the electrons to drive the synthesis of polymers of 6 carbon sugar from carbon dioxide and water molecules. The overall balanced equation is written as 6 carbon dioxide plus 18 ATPs plus 12 NADPH plus 12 water molecules are required for the synthesis of single molecules of glucose, and 18 ADPs, 18 aromatic phosphate, 12 number of NADP and 6 number of protons are prepared. The reactions that generate the ATP and NADPH used in carbon fixation are directly depends on light energy. Thus, stages first to third are called the light reaction of photosynthesis or light dependent phase or Hill's reactions. The reaction in the stage 4 are indirectly dependent on light energy. It is known as dark reaction, or it is also known as Blackman's reactions of photosynthesis, because they can occur in the dark utilizing the supply of ATPs and NADPH generated by light energy, that means which is synthesized during the light phase of photosynthesis. And now these ATPs and NADPH are utilized during the dark phase of photosynthesis for the conversion of carbon dioxide to carbohydrates. Thank you.